Okay, so the next speaker is Elisabetta Spinazzola, and she will talk about metadata management and the storage. So Elisabetta, please.
Good morning, everyone. And now I will share my presentation about metadata management in biomedical imaging, as far as the metadata management and the storage. From my side, we have before to speak a bit about three main concepts, which are FAIR principle, metadata, and ontologies, to then see what has been developed by my side. Conventionally, the research data lifecycle comes from the data collection, acquisition, then go through the process of the data, and then they will be published. In general, this data will go in software, images, documentation, and in a really few cases, they go in publication. So this makes the data not more discoverable in the future, and integrated and analyzed. So the idea is to introduce two more steps that can curate the data and archive them in server or repositories or more publication to make the discover and then the analyzation. The idea is to find this four data principle of FAIR, which means findable, accessible, interoperable, and reusable, to include such many kind of data, such as data, metadata, images, softwares, web service, repositories, to conclude the life cycle of the data and make the data more findable, which means discover them in a repository, in publication, in an easiest way. Then accessible through a license, so we can access and see the data in a easiest way. And the accent should be post on the interoperability and the reusability of the data. Since there are a lot of data and the amount is very huge, the reuse of this data could be very useful, and also the interoperability between different resources. So why we should adopt this principle? Because they facilitate the sharing of the data, and as we said, they reduce the cost of the duplicating data collection, and also allow the data usage to cross disciplines. So what does the metadata? So the subject to apply the FAIR principle, they are information of different kind of types that describe, explain, locate, classifies an information resource, and they have been created to describe in a standard way the data. So they could be of various types for different application as far as the research, and in particular, in our case, for preclinical studies, but in general also scientific study, especially for the standardization of a systematic management, analysis, and storage, which could be, of course, better to apply the FAIR principle. Although there are a few examples of metadata models, there is not a standard of metadata model or list, especially in preclinical studies. The few examples that we have are, for example, the REMBI, which is the recommended metadata for biological images, guidelines for the scientific community to standardize the dataset annotation. Then we have the Arrival Essential 10, which are requisite for publish some data set and data, which make also the research more affidable. Then we have an initiative from a project from the NIH, the CIMI, which has made this project about the integration of DICOM standard with the co-clinical data. Now let's talk about ontologies, which are a language and a representation of the knowledge of a certain domain. We have terms that are classified in a specific domain, and each domain is defined a semantic or the meaning of the terms in a both way, human readable and machine computable. So we have classes and relation through URI or IRI. Then we have the domain of the vocabulary, and this is applied to metadata description. We talk about linked data to represent a method of publishing and sharing the data on the web, as we said, the URI. And there are several repositories that we can find on the web to choose and use ontologies of different domains, such as the bioportal, the ABI, and a lot of links comes from the biontology. A briefly view about the format that we have for the ontologies. We have a graph-based language for RDF, which is a framework XML, then a OBO flat file format, and then a script code of UL web ontology language, which can be interrogated with SparkQL language. So the aim of our project is to create a metadata model for preclinical imaging dataset, referring in particular to the application of FAIR principle, the cross-researching between different resources, and of course infrastructure, and then standardization, especially standardize the description and the curation of a preclinical imaging dataset. The metadata that in this case we are talking about are a set of data information that allow to describe and give detailed information about preclinical imaging dataset, as far as animal demographics, study protocol, biological, medical, and chemical characterization, and of course imaging modality. As we can see in this picture, we have a different group of dataset, which depends on the kind of the studies, and all this data have to be standardized and organized in a particular way, since also the amount of data is very huge. In our case, the model comes from the union, the merge of the two examples that we've seen before, alias the REMBI and the ARRIVE guidelines. At the first, we had just 50 entries of different domains, biological, chemistry, and imaging. Then, with the Euro-BioImaging working group of data management, we have applied this list to 150 entries. And now we are trying to sharing this metadata list to have more evaluation and consensus among both Euro-BioImaging nodes with the data management working group and also other researchers. The current metadata model is divided in 8 sections and many of them as a subsection. As we can see here, just a few examples, but then we can see it in more detail. The study design is composed of study design and publication, which report all the information about the publication of the studies. The study component with the imaging technique and dataset information. EF, in vivo experimental parameters, is composed of study design, sample size, exclusion criteria, a lot of information that has been standardized to be as more complete as possible. Then image acquisition, data, correlation, and analyzed data for a total of 174 metadata. As we can see here, other section, for example, experimental procedure with the subsection, and also EVEP, which is a very big part. Then what we have do, we have selected some metadata from different domains. As we can see, drugs, so anesthesia, pharmacological drugs, contrast agent, then imaging modality and submodality, disease model, species, organ tissue as anatomical parts, strains, gene, and cell line, to treat them with ontology and curate this data. The ontology that we have chosen are NCIT, which is a very big ontology with lots of terms of different domain. In fact, we use this ontology for different domain as imaging modalities, gene, anatomical part. Then we have used KB for chemical entities. CLO is for cell line, and OID is for disease. The final goal is to make the research easier with the use of synonyms, as we can see later with the brief example, which are coded by the ontology. The main ether for this construction project is the identification of the ontologies between a lot of them. And so here you can see the link between the metadata domain and the ontology that we have chosen. Then we have stored the information of the ontology terms that we need to describe our metadata for our dataset in a table. And then we have made the link between metadata terms and ontology with a univocal code. For example, we have this dataset 0001, which has as anesthesia drug disoflurane. This terms is identified by the chemical ontology entitized by a code, which is 6015. We apply this code in another metadata, which is KB anesthesia, and this linked gives us the opportunity to call disoflurane also in another way, if is of course mentioned by the ontology. Meanwhile, we do the research. The storage of this information has been done with the construction of a database. We used the PostgreSQL, and then we have inserted all the information in table through an SQL language. We have made so the PIDAR, which is a repository for preclinical imaging dataset, and we have started it last year. We are continuing collecting a preclinical imaging dataset with data curation, and our aim for the future is, of course, to continue doing this and make a survey across several preclinical imaging centers to define and have the consensus for our model, metadata model. We also think to write a consensus paper, white consensus paper, for share our preclinical metadata model involving also the participant of the survey. Thanks for our attention. Now, if you have some question,